I'm going to introduce today's speaker. We have Gelalcha. He is a postdoctoral research associate here at the University of Tennessee in the College of Veterinary Medicine. So I'm going to turn it over to you to take it away.
Thank you so much, Aliza, and good afternoon, everyone. And thank you also for coming. And it's my pleasure to walk you through part of my PhD work focusing on antimicrobial resistance and molecular epidemiology of extended spectrum beta-lactamase producing E. coli in East Tennessee dairy farms. So to start with introduction, antimicrobial resistance is one of the most urgent and complex global health challenges that we are facing today. And globally, bacteria-related antimicrobial resistance was associated with 4.9 million deaths in 2019 alone. And according to CDC, in the United States alone, antimicrobial resistance accounts for more than 2.8 million infections and causes over 35,000 deaths per year. And this figure is only expected to rise if effective intervention is not put in place. According to a report by O'Neill, estimated that by 2050, antimicrobial resistance could kill 10 million people annually and causes. And this figure could surpass even cancer, which is the main cause of death currently. And with human health impacts often receive the most attention, and it's associated with various figures. But the impact of antimicrobial resistance goes beyond human health. In agriculture, particularly in food-producing animals like dairy, cattle, antimicrobial resistance contribute to treatment failure, increasing disease burden, lower productivity, increased culling, and significant economic loss. And so the impact of antimicrobial resistance is a textbook example of one health issue that's affecting animal, human, and also environment alike. So if we look at the drivers of this antimicrobial resistance, the main drivers of antimicrobial resistance is selection pressure, which is caused by antibiotic use, both in human and animal setting. In livestock, we use antimicrobial just to treat sick animals, also for preventive purposes, and sometimes also metaphylactically to control infection in a group of animals when some animals are showing symptoms. In human, antibiotics are typically used for treatment, but also sometimes for prevention, especially in immune-compromised patients. So when misused or overused, these antibiotics select for resistance bacteria, which can spread within a species and between species. So when bacteria and the resistance gene can move between human and animals and the environment. So if we see resistance bacteria in animal setting, there is a risk in the human health setting and vice versa. So the impact in one sector can inevitably affect the other. So today I'm particularly focusing on a specific group of antibiotics and the resistance against them, specifically beta-lactam antibiotic, which is widely used both in human medicine and also in animal production. These beta-lactam antibiotics are actually characterized by the beta-lactam ring, which you can see as 3-carbonamide ring, which is considered as the functional group for the beta-lactam antibiotics. And it is the structure that goes and binds to specific parts of the cell wall of the bacteria and prevents cell wall production and kill the bacteria. And the bacteria also targets the same beta-lactam ring. So when bacteria develop a resistance, target this beta-lactam ring, and so it breaks down the antibiotics and renders it harmless. So in US, these beta-lactam antibiotics, particularly in dairy farms, are most frequently used antimicrobials. It's particularly cephalosporin, like ceftiofur, called also third-generation cephalosporin or extended spectrum cephalosporin, and also cefaprin, which is first generation cephalosporin, and also penicillin. They are commonly used in dairy farms, mainly for the treatment of mastitis and also other reproductive gastrointestinal diseases and respiratory diseases. So here, the widespread use of this cephalosporin could select for beta-lactamase producing bacteria, a bacteria that could produce an enzyme called beta-lactamase and that target the beta-lactam ring and make them useless or not effective. Particularly, this beta-lactamase, the third generation cephalosporin, actually selects for the extended spectrum beta-lactamase like ESBL. ESBL stands for extended spectrum beta-lactamase and is produced mainly by E. coli and also several other group of bacteria, especially those in the group of Enterobacteriaceae. So the concern here is the same types of bacteria, the same types of antibiotics are used in human health settings, like ceftriaxone and cefotaxime. There are third generation cephalosporins, which are categorized as critically important class of antibiotics, and they are reserved for the treatment of severe infection in human, including sepsis. So if we are using a similar antibiotics, both in animal setting and in human setting, the resistance that is created, for example, in dairy farm, can cross into the human health setting, particularly through different routes. It could be through direct contact or through food chain or indirectly through the environment. And similarly, the resistance that is created in the human health setting can also go to human. So nowadays, what we are seeing, according to the CDC report, is a rising increase in the number of infection caused by bacteria which are producing extended spectrum beta-lactamase, particularly the Enterobacteriaceae bacteria. And from 2012 to 2017, there is a continuous rise in the number of infection and hospitalization caused by this group of bacteria. And annually, there is over 9% increase in infection, that according to the CDC report. So traditionally, these ESBL-producing Enterobacteriaceae were mostly hospital-acquired infections. But in recent years, what is being observed is a significant epidemiological shift. Nearly half of these infections are reported to be community associated. That means this infection is present in the community or in individuals that have no prior exposure to hospital or have not taken third generation cephalosporin, which could select with this group for this group of bacteria. So this epidemiological transition suggests the presence of additional reservoirs beyond hospital environments. One of such likely contributor is agricultural use of critical important class of antibiotics like third generation cephalosporin, which is the veterinary analog, the human analog for ceftriaxone or cefotaxime. So while we cannot draw a direct cause-effect line from farm use to hospital infection, but it's reasonable to look beyond hospital walls and examine what's happening in animals, such as dairy farms. So despite the widespread use of this beta-lactam antibiotic in dairy farms, there is limited information on the prevalence of ESBL coli in dairy farms, especially in Tennessee and generally also in the US. And there is also lack of information on the factor that can contribute to their occurrence. We don't have also a lot of information on genetic diversity and the mechanism of spread of this bacteria and their resistance gene, And addressing this gap is important to inform risks arising from dairy farm and devise evidence-based effective control measures. So we have three objectives And the first one is to determine the prevalence and antimicrobial resistance profile of extended spectrum beta-lactamase E. coli, and then characterize the genetic diversity and the mechanism of spread of ESBL gene spread. And the last one is to look at the predictors or the factors that contribute to the occurrence of this extended spectrum beta-lactamase producing E. coli and identify potential control points within dairy farms. So to achieve this objective, we used a cross-sectional study involving about 40 dairy farms in East Tennessee, and we collected about 508 rectal fecal samples from dairy farmers and 64 farm environmental samples, which we can see in the next slide. And we also administered just brief questionnaire survey to look at the general farm management, farm antibiotic use pattern, and some animal level information. So, as I said, we collected a total of 572 samples and about 424 rectal fecal samples from cows and 84 from calves. And also from environmental sample, we collected 13 manure samples, 19 water from directly from the feed trough, from the water trough, and feed also from the feed trough. And then for bacterial isolation, we prepared the sample and we directly inoculated into Chromagar ESBL plate, which is a differential and a selective media which only selected extended spectrum beta-lactamase producing bacteria. And E. coli, they appear as dark pink and reddish colony on the medium. So the Chromagar ESBL plate gives us the preemptive ESBL colony, and we confirm that one using MALDT of for E. coli. Then E. coli confirmed bacteria were antimicrobial susceptibility, subjected to antimicrobial susceptibility testing. And we took pure E. coli colonies from blade agar and transferred, inoculated into a 96 wear titer plate. And then this titer plate contains about 40 antimicrobials with various antimicrobial concentration gradients. And then we checked if we looked at the growth of the bacteria in the presence of this antimicrobial resistance, antimicrobial agents, and interpreted the value depending on clinical laboratory standard institute interpretation criteria. Then we also, from all AST tested E. coli, we extracted genomic DNA using MarkTract DNA extraction kit. We prepared the library using next era library preparation kit, and we sequenced and we generated paired-end FASTQ files. So we used combined statistical and genomic data analysis. We used both descriptive and inferential statistics. Descriptive statistics to summarize various prevalence and resistance profile at the farm and individual bacteria level. Also, we use mixed effect logistic regression analysis to identify the risk factors. And we use several bioinformatic pipelines from quality control pipeline like FastQ, MultiQ, and Trimomatic up to downstream analysis on assembled genome. So to start the result from the farm management and survey, half of the farms practice closed production system. That means they rate their own replacement stock, and half of them are introducing new animals. And 75% of the farms discard waste milk from cows on antibiotic treatment, and while the rest, about 29%, are feeding to dairy calves. And about 12 of the farmers practice blanket dry cow therapy, where they treat dry cows at the start of the dry period and irrespective of the risk status, which is also one of the major risk factor for antimicrobial selection pressure. And we identified that mastitis the most frequent health problem. And again, beta-lactam antibiotics, especially cephalosporin, both first generation and third generation, and penicillin were the most frequently used antibiotic in dairy farms. And this slide is we are looking at the prevalence of ESBL coli by sample source. We isolated a total of 233 E. coli isolate, which is making 40.7% overall prevalence of on overall prevalence to the sample level. And we identified manure was 46% of the manure also contain E. coli, and rectal fecal samples 41% of rectal fecal sample also has E. coli, and the prevalence in feed was 33%, whereas that in feed was 26%. And generally, we know there is no significant difference in prevalence among the different sources, indicating that the bacteria is widespread in the farms. When we look at the prevalence pattern of E. coli across the farms, both individual animal and between the farm, the weighted fecal prevalence of ESBL E. coli was 47.5%. Nearly half of the sampled animals were shedding ESBL E. coli, and almost all the farms, not almost all the farms sampled have at least one animal which is shedding ESBL E. coli. And the within farm prevalence of ESBL E. coli ranges from 8 to 100%. And this variation is statistically significant. So the lowest one is 8 and the highest for 100% when we checked all sampled animals were positive. We also looked at if there is a difference between prevalence of ESBL E. coli between cows and calves, and we found that there is statistically significant difference between the prevalence in cows and calves are almost twice more likely to shed and carry ESBL E. coli compared to dairy cows. And similarly, we also looked at we looked at several factors, but these 3 factors were turned out to be independent risk factor, which were associated with cow level and also antibiotic use related risk factor. We compared ESBL prevalence between cows which were recently treated with CFT04 and which are not treated, and cows which are recently treated with CFT04 were not surprising, significantly higher in shedding ESBL E. coli compared to those which are not shedding. Again, lactating cows, the prevalence in lactating cows were significantly higher compared to the dry cows, and cows with high parity have also significantly higher prevalence compared to lower parity cows. When we look at the resistance profile of ESBL E. coli across the 40 antibiotics tested, the resistance is extremely high to extended spectrum beta-lactamase. Actually, it's no surprise because our media also selected for those ESBL producers. So nearly 100% of E. coli were resistant to ampicillin and ceftriaxone, which is third generation cephalosporin. And so these two are just the hallmark of ESBL activity. And in addition to the beta-lactam antibiotics, the E. coli also displayed resistance to multiple classes of antibiotics, especially to tetracycline, sulfadoxazole, and streptomycin. And again, there is also low but notable level of resistance to critically important class of antibiotics like gentamicin and naldixic acid. And likely, there is no resistance against meropenem, which is considered as one of the last resort antibiotics used in human medicine. We also looked at the distribution of multi-drug resistance E. coli by sample source, and the majority of the samples, for example, 100% of E. coli isolate obtained from manure and water were multi-drug resistance, and 93% of E. coli isolated from cows and 96% of E. coli isolated from cows were multi-drug resistant. So overall, the multi-drug resistance E. coli, 94% of E. coli isolate were multi-drug resistant. And moreover, about 40% of the isolate will concurrently resistant to at least 6 different classes of antibiotics. But when we compare the multi-drug resistance prevalence between the different sample sources, they were not statistically significant. So it's widespread across the farms. We also looked at the distribution of beta-lactamase gene in ESBL E. coli, and out of the 233 sequenced E. coli, isolate 98.7% of them carry beta-lactamase genes. And the most predominant one was beta-lactamase, CTXM variant, followed by TM, which is carried by more than half of the sequenced isolates. And we also detected other class of beta-lactamase, including the beta-lactamase lab2, which was previously only reported in human in the US, and there was no reports from dairy farms in the US before. So in addition to these are the BLA genes are normally they are plasmid mediated resistance gene. We also detected the chromosomal mediated beta-lactamase gene, and about 39% of E. coli isolate have mutation in MC gene promoter, and this mutation also leads to constitutive expression of beta-lactamase from the chromosome, and which also causes beta-lactamase activity. So when we look at also other resistance gene, we found 97% of ESBL E. coli were also genetically multi-drug resistance, which also concurs with the phenotypic observation that we had. Nearly all isolates carried fluoroquinolone resistance gene. As you can see, about 229 carry different kinds of fluoroquinolone genes, and tetracyclines are also one of the most commonly observed resistance genes, which are co-carried by beta-lactamase genes, and followed by folate pathway inhibitors like sulfonamides and aminoglycoside, chloramphenicol, and macrolides. Especially macrolides is also considered as one of the critically important class of antibiotics. So a significant proportion of E. coli isolate from animals are carrying resistance gene to this group of bacteria. When we look at genetic diversity and the spread mechanism of ESBL E. coli, we observed about 47 distinct sequence types of ESBL E. coli, and including 4 novel sequence types. And most ESBL E. coli sequence type exhibit clustering within farming. We will see in the next slide. So most isolate within the same farm have a kind of similar sequence types, indicating there is animal-to-animal transmission or clonal expansion of this resistance gene. And we also detected identical sequence type, resistance gene patterns, and resistance profile in different farms, and which also indicate potential transmission between different farms. We also detected a matching ESBL sequence types between environmental samples like water, feed, and fecal sample on the same farm. So on this radial phylogenetic tree ESBL E. coli, we can see at least 4 distinct information. And first, we can see the within farm clustering of the isolates. If we see here the first those isolates which are named with the same prefix, the same first 2 letters, they have the same origin. That means they're obtained from the same farm. So you can see, for example, here for AG, about 14 isolates, they are clustered together. They're on the same branch of this phylogenetic tree, and indicating that there is clonal expansion or animal to animal transmission within the same farm. And similarly, you can also see cross-farm clustering. If you see here, there is, in addition to the AG, you can also find the DG and DG5, DG8. And these are different isolated obtained from different farm, but within the same county. And they are also closer together. These farms are also closer together, and they do have the same resistance pattern. And again, you can also see another group of isolate obtained from a different farm having clustering together with other isolate obtained from two different farms. So you can find this clustering across all these. For example, this farm all are RM, so they are obtained from the same farm, so they are clustering together, indicating mainly this bacteria is spreading through clonal expansion within the farm. So in addition to this one, we can also see clustering of isolate between the rectal fecal samples, between feed and water, as in these two. So within the same farm, you can see the same sequence type or the same clustering or appearance on the same branch of E. coli obtained from water, from feed, and from also manure, indicating that there is also this environmental, the environmental samples are also varying the same, so they can serve as a continuous source or maintenance of this bacteria within the farm. So we can, so in addition, what's other most important thing that we find is 28% of these isolates were high-risk E. coli clones. That means they are potentially pathogenic. And especially, we find the sequence type 10 and sequence type 69. These are identified from clinical cases in human in the US or also across the globe. So the detection of these two high-risk clones of bacteria suggested that dairy cattle might serve as a reservoir of clinically relevant strains, especially where about 64 of these bacteria are categorized as high-risk clones. So in a conclusion, the prevalence of E. coli in dairy farm is high. We find nearly half of the animal in the farm, nearly half of the sampled animals are shedding ESBL-E. coli, and almost at least one animal is ESBL positive across the farms. So it is very high and widespread. And treatment with third-generation cephalosporin lactating cows with higher parity and calves are independent risk factor for ESBL-E. coli. So these are potential area where we can do a targeted intervention measure. And most ESBL-E. coli are multi-drug resistance, both phenotypically and genotypically. And if these bacteria jump into, or if they can cause clinical disease, either in animal or in human, so the treatment is going to be very challenging. And beta-lactamase gene appears to spread via clonal expansion also, or via horizontal gene. Identical sequence types and resistance profile across the farm. This detection of these identical sequence types and resistance profile indicate possible interfarm spread or transmission. And detection of matching sequence types and resistance gene between fecal, manure, feed, and water samples indicate that these samples can serve as a source or reservoir of ESBL-E. coli in the farms. So most importantly, nearly 1/3 of ESBL-E. coli sequence type belong to high-risk clones. So as I said previously, dairy farms could serve as a reservoir of these potentially human pathogenic multi-drug resistance E. coli isolates. So finally, I would like to thank my PhD committee members at Department of Animal Science who provide me and support and helped me during this study, all graduate students who helped me with collecting the samples and also running some of the analysis. Tennessee dairy farmers who allow me to work on their farms, UT diagnostic bacteriology and mycology lab where I run antimicrobial susceptibility testing and the MALDI-TOF and UT Genomic Core Center. Thank you. And with this, I will take questions.
Thank you, Dr. Gelalcha. That was very informative and pretty alarming as well. If anyone has any questions, you should be able to unmute yourselves and ask in person, or you can also put them in the chat as well. We'll just give everyone a few minutes.
Dr. Benti, I have some questions for you.
Okay.
Thank you for the very insightful presentation. And I think maybe you will answer one by one because there were quite many as you presented that I had noted down.
Okay.
The first one is on the prevalence of ESBL that was reported. What approach did you use to come up with this prevalence? Because at one time you use Chrome agar, and then you go on to do the other molecular tests. So is the approach of how you come up with your positives, was it based on parallel testing or on serial testing with different methods?
Yeah, thank you. So I will answer the question and then you go to the next or
Yep.
yeah. For determining the prevalence, I use Chrome agar ESBL plates. So this Chrome agar ESBL plate actually grows only those bacteria that are ESBL producing. So it selectively grows those which are ESBL producing and also differentiates the bacteria. For example, if it is E. coli, it is dark red or it gives you dark red or pinkish color. And if it is Klebsiella, Enterobacter, or Citrobacter, it gives you metallic blue color. And then from this, you take the E. coli isolate, and then I go and do whether it is E. coli or not using MALDI-TOF. So in short, for the base is the growth on ESBL E. coli, or on ESBL Chrome agar.
I see, thank you. Because I needed really to understand the sensitivity and specificity of relying on Chrome agar as much of our. In this case, I think you used it as your screening and confirmatory test, and at least you've told me that it quite grows only the ESBL, but again, within the culture itself, there are other colonies that are growing in there. So, how really sensitive is it and specific is it?
Good, thank you. Here is the thing. So normally Chrome agar ESBL, it's 98% sensitive and 97% specific. This has been established. And this is also what I see from the genomic sample, from the whole genome sequence analysis, and also from antimicrobial susceptibility testing. So if you see in the one of the slide, 98% of those E. coli isolate, they contain beta-lactamase genes. That means it also confirms back what I'm saying. It has 98% sensitivity and 97% specificity. So they would grow. You may see several colonies on ESBL Chrome agar. But you differentiate those based on the color, and you go back and also do confirm the species of the bacteria. And in my case, I don't have much concern because I did Yes, antimicrobial susceptibility testing. And again, I also have whole genome sequencing result, which can look at whether the expressed phenotype is supported by the presence of a genotype. But one thing here, your question is valid. Normally, to look, the standard protocol to look at whether a bacteria is ESBL producer or not is double disc synergy test.
Right.
Yeah, it's double disc synergy test. But in this case, because I'm looking from end to end, I don't want to do double disc synergy test in all those isolates because it's very costly.
All right. And then why was really what was the whole intention of trying to study multi-drug resistance in a bacteria which has already been classified as multi-drug by a couple of AMR regulatory bodies?
Okay, you mean?
Because the results are already showing you by them already being 98% multi-drug resistant. So what was the whole intention of studying again multi-drug resistance in a phenotype that has already been almost classified as a multi-drug resistant strain by the WHO and other bodies?
The 98% multi-drug resistant that I'm talking is just my result.
Oh, I see.
It's my result. It's not from other bodies. So we said the sensitivity and the specificity of Chromagari ESBL is 98% sensitivity, 97% specificity. I said I confirm also this same from my from the whole genome sequence analysis because 98% of them contain beta-lactamase genes and they also carry other resistance gene.
All right.
Yeah.
Then the other question is regarding is much more of a systems dynamics question.
Okay.
So in your results, it shows that they are quite most of these animals were really much more carrying high numbers of ESBL, and then the same and this is within a setting which is quite having top-notch policies and systems, that is the US. And then
Yeah.
when you compare that result almost with settings much more in the L-mix countries, you find that it's almost the same result where you're having also a couple of these farm animals in the L-mix settings also carrying high levels of ESBL. So in this case, what really can be done to reverse AMR? Because we thought that by having some of these better systems and better policies to drug access as it is in the US, it could be reversed in another setting, but it's almost now the same picture.
Mm-hmm.
Yeah.
Yeah. So the thing is, even if you see, if I talk in some, for example, in Ethiopia, if you take about developed country, for example, if you take the case of Ethiopia, the farmers, they don't have much access to third generation cephalosporins. They have a lot of access to but other beta-lactam antibiotics. So it's the reason that we are finding very high prevalence of ESBL producing here, particularly in intensively producing countries like the US, is mainly also related to intensification. Also comes with its own cost. There is high disease pressure. And again, especially with the dairy farm, the third generation cephalosporin, they are much preferred because the withdrawal rate for the milk is almost close to zero. The farmers are not losing milk when they are using that antibiotic. So they often use this third generation cephalosporin, and this also selected for ESBLs. But as you said, the prevalence is a kind of the same. Particularly, you see the human burden of antimicrobial resistance. It is even higher in developing countries, particularly in Western Sub-Saharan African countries. It's mainly related to also the infection pressure. We have high level of infection, and we also use high level of antibiotics, and particularly related to also hygiene and all other those kind of things. So the solution normally it comes to, if the selection pressure arises from the use of antibiotics, it comes to reducing the use of antibiotics or having stewardship towards antibiotic use. How do we do that one? It goes to reducing disease burden, focusing on prevention, focusing on biosecurity, focusing prevention, particularly, for example, if the disease is vaccine preventable, using vaccine, so that we don't have to treat animals and export. Treating animal is always there, so those kind of things, and also reducing the overuse of the antibiotics. Sometimes we use antibiotics as an excuse for to complement, or at least to not to complement, but to replace other activities that we should use to reduce this burden.
Yeah, thank you.
You're welcome.
Does anyone else have any questions? It looks like we have one in the chat. Michael Maharo asks, what factors were associated with cross-farm transmission of resistance isolates?
Okay. Okay, it's a good question. So we are in this study, we were not able to look at the factors, but we can speculate, especially between two of the farmers which are adjacent to each other in this farm. So what we may think is, for example, movement of personnel between the two farms, which might carry the bacteria from one farm to the other. It could be also use of shared vehicle, especially most of the farmers they use trucks to spread the manure into the farm. They use manure as fertilizer if they're sharing the same vehicle. And again, there could also be, for instance, some animal reserve animal vectors which can move like rodents. They are moving between different farms. So this could be some of the things. And again, 50% of the farmers, we said also, 50% of the farmers introduce new animals. They introduce new animals from other farms. So this can also bring the bacteria with itself, and which might be responsible for inter-farm sharing of the same sequence types of, or the same identical bacterial strains between the different farms.
All right, are there any other questions for Dr. Gelalcha? If not, we may give everyone about 10 minutes back of their day by finishing a little bit early.
Yeah.
I don't see any more in the chat. All right. Well, thank you very much for that presentation. That was really interesting. If anyone's interested in watching it again, it'll be up on our website later this afternoon or by tomorrow morning for sure. On our seminar page of our website, you can see all of our previous seminars there. Thank you again for joining us, everyone, today, and thank you very much, Dr. Gelalcha, for making time to share that research with us. That was really
You're
interesting,
welcome.
and we appreciate
Yeah.
it.
Thank you.